Thanks, Charlie. I'm a professor at UNC Chapel Hill, and I'm delighted to introduce these Ian and Jess. So Ian received his BS from the University of Virginia in 2016. After that, he spent a year at Duke working with Charlie, actually, a very productive year, yielding some papers. He's been a PhD student at Princeton with Jess since 2017, and he's published an impressive 8 papers as first author. So Jess has produced, well, backing up. So Jess got her BA from Oxford in 2000. She got her PhD from Imperial College London in 2005. She's been on the faculty at Princeton since 2014, and she's now an associate professor. So Jess has published a truly astonishing number of papers, now that I try to look them all at once. But more importantly, I think both Ian and Jess have published an amazing diversity of research topics, ranging from evolution to ecology to policy, and in systems ranging from humans to plants. Even more impressive than that diversity, though, I think, through Jess's career, is that the diversity is united by a common conceptual and methodological thread of demography. She has shown how demography and approaches that are rooted in it, like SIR models, can be applied to further our understanding and inform policy across really an amazing breadth of science. And today, I'm delighted that they will be discussing their recent application of such models to an issue of tremendous current importance, the risk of vaccine-driven evolution in SARS-CoV-2.
Well, thanks so much, Charles, for that invitation, and thank you so much for that introduction, and thanks so much for the invitation. It's a great honor to be here. This is work that was very much driven and inspired by Ian's brilliance, but I am going to do the presentation, and then he will hopefully answer all of the questions. I think that's the way this will work best. And we wrote this paper, as Ian was pointing out, as we were discussing it at the start, right in the sweet spot where we knew just enough to be able to think about what the future might hold, but it wasn't so incredibly confusing. It was slightly pre-Omicron. So many things have changed since we wrote that paper. Just to give broad context, I know that everyone's been thinking about this sort of probably it's been very hard to avoid, but the way that SARS-CoV-2 works is to be infected, you're exposed to the virus, which binds to ACE2, maybe less so for Omicron, enters our cells, starts to replicate within our upper and lower respiratory tract. And then hopefully one outcome is it's cleared by immunity. You might have worse symptoms associated with replication lower in the lungs, and immunity also drives many of the symptoms. And clearly from this premise, from this basic biological trajectory, there has been a whole sea of evolution that has happened to this virus. And it is a sea of evolution that increasingly concerns us. The things specifically that concern us are phenotypes associated with increased transmissibility, so moving faster from individual to individual, increased capacity to overcome natural or vaccinal immunity, because then we have no protection, and then increased virulence. These are the three, so the virus is probably changing all the time, but these are the 3 ways that matter to us. A really key point in thinking about the evolution of virulence is that virulence alone is never going to increase the fitness of the pathogen, right? It's just, it has no fitness benefits if it is not associated with some other trait. But for many pathogens, it does seem that there is potential for host damage to enhance transmission. So for example, it might induce coughing. And I should also say, the word virulence usually refers to damage to the host. So increases in morbidity, increases in mortality. If one does have a situation where virulence is associated, for example, with an increase in that transmission rate, then obviously it could be in the interests of the virus, or it could be an evolutionary advantage for the virus to increase its transmission. Sorry, to increase its virulence, because then that would increase its transmission. But, and this has been known for a very long time, in this scenario, if one of the things that virulence is doing is removing infected individuals from the population, perhaps because they die, then although you might be increasing per time step transmission, you would be reducing the total time during which transmission could occur. So you're increasing transmission rate as virulence goes up, but you're reducing transmission rate, so it's transmission time as virulence goes down. An important thing that's probably popped into your mind here is that if we think about SARS-CoV-2, many of the most severe symptoms happen long after transmission has occurred, it seems. The majority of individuals experience morbidity or death several weeks after the initial infection. So if such a trade-off does exist, if there is a virulence transmission time reduction trade-off for SARS-CoV-2, it's likely to be associated with increases in symptom frequency, which might encourage people to isolate themselves or might put them in a state where they're much less likely to mix. If these two things hold, what we expect is that because virulence increases transmission per time, but reduces the total time available for transmission, you end up with an intermediate optimal. So there is a virulence level at which the virus is expected to optimally transmit. And if we take just this very basic, sort of very qualitative sense of the world to SARS-CoV-2, the two questions we might want to ask is, one, is the evolution of increased transmission possible? And then, is increased transmission linked to increased virulence? So what do we know? Is the evolution of increased transmission possible? Well, the answer seems to be yes. Alpha and Delta and now Omicron do seem to be more transmissible than the ancestral strain. So an unmitigated yes, we can definitely have increased transmission. And in fact, it's sort of quite astonishing that every time we've had a new strain, R naught seems to have jumped by a factor of 2, which is really amazing. I don't think we've seen that elsewhere. So the second question is increased transmission linked to increased virulence? And the answer there is sometimes. So it does seem that Alpha and Delta were associated with greater virulence. So for example, Alpha was associated with a 55% increase in the infection fatality rate, and Delta possibly too, but it's sort of the evidence was more mixed. And there is, of course, the complexity that's arisen more recently of Omicron. It is very hard to say exactly what Omicron is doing compared to the ancestral strain, because we are in a world where there's an awful lot more immunity in the population. So it's very murky trying to figure out what Omicron precisely is doing. And for example, the really severe outcomes in Hong Kong suggest that, you describing Omicron as mild might not really capture precisely what's going on. But just looking at it, it does seem like Omicron increased transmission and didn't, if anything, at least on that particular immune background, reduced virulence. So the key point is that we do know that transmission can increase. We don't know if there is a relationship between virulence and transmission particularly. It's not beyond the bounds of possibility. And when we started working on this paper, we thought that although current evidence, the data at the time, and in fact the data now, was still not enough to conclusively determine that there was such a trade-off, or even if there was such a trade-off where SARS-CoV-2 fell, we thought it was worth starting to think about this, in particular because we were really interested in the fact that there was a massively changing landscape of immunity. And one of the ways in which the landscape of immunity, which will drive selection on the variant, was changing was through the glorious deployment of vaccines, which was such a delightful occurrence in 2021. And if you start thinking about vaccines, you need to start thinking about how vaccines might also shape this relationship between virulence and transmission, and what consequences this might have as well for the evolution of virulence. And of course, it will hinge fundamentally on how vaccination intersects with this underlying biology. This is something that we've known since Sylvain Guindon wrote a lovely paper on this topic, showing that if vaccines reduce symptoms, so they reduce that virulence, and there is a relationship between symptoms and transmission, you can actually have selection driven by vaccines for increased virulence. Ian, in his brilliant way, identified that this was actually quite a relevant question in thinking about SARS-CoV-2 vaccination quite early on, or potentially relevant, because you can think about infection as being, so this is perhaps a cartoon of the way it actually happens, but it might be that infection in the upper respiratory tract was milder, infection in the lower respiratory tract, to a first approximation, is more severe. And it does seem that natural infection provides protection in both compartments, but early vaccine trials suggested that vaccination was really only providing protection in that lower compartment. which aligns also with our experience of vaccines preventing severe disease, but being less successful in preventing transmission. So there is potential for this sort of scenario to go on. And it is a burningly important question, of course, because of many reasons, but in particular because of global inequity in vaccination coverage. So what we decided to do, or rather what Ian led, was to put together a really simple model of how these different compartments, how if we think about upper respiratory tract protection as separate from lower respiratory tract infection protection, might shape the evolution of virulence in SARS-CoV-2. Again, bearing in mind that we don't have a strong premise for knowing that there is actually this association between the two, it's very possible that, you know, even if such a trade-off exists at some scale, we might still be very far from it. The virus might have a lot of wiggle room before it ends up butting up against those limits. Ian built a very simple model that essentially had both transmission and virulence associated with two different compartments, an upper compartment, and there was differential protection associated with vaccination in the two. So then you can ask what would happen with different levels of impact of the vaccine in terms of protection of the lower respiratory tract and the upper respiratory tract, with the upper respiratory tract having a greater contribution to transmission. And it's actually, you know, it's relatively straightforward to see how this is going to work. So on the y-axis here is lower respiratory tract protection, and on the x-axis is upper respiratory tract protection. And what we expect, based on Sylvain Guindon and huge amounts of work on this topic, is that as lower respiratory tract protection increases, if there is these sorts of patterns, these sorts of trade-offs are in play, then we expect selection for increasing optimal virulence. Conversely, if upper respiratory tract protection increases, we expect declining incidence with no particular cost or benefit to virulence if that's all that's happening, and perhaps progress towards herd immunity. And this is indeed what that basic model shows. So the colors here show you the trajectory towards, the optimal virulence that emerges from this model, so the virulence that maximizes fitness. And as you move up in terms of lower respiratory tract protection, this is moving between 0 and 1, sorry, the y-axis and x-axis aren't labeled, you end up with more virulence. And not shown on this plot is you can even end up with, well, actually it is shown. You can even end up with selection, unbounded selection for high virulence. The gray area just shows you that it's just going to get worse and worse and worse. But you can make things better by increasing upper respiratory tract protection. And of course, the bind here is that, of course, we want lower respiratory tract protection. This is the thing that, this is how vaccines are helping us. The complexity is that it can have this, this evolutionary outcome that is something that we're pushing down the road. And again, this all hinges on the particular patterns of the biology that we don't necessarily have a perfect handle on. And it also depends on what assumptions you make about what proportion of the population is vaccinated. So this is just showing results for 75% vaccinated. If you up the proportion vaccinated to 90%, then you get a greater area where there's actually evolution-proof herd immunity. You're actually driving the virus into a place where it just can't persist at all. There's an area where the virus cannot get off the ground. So its effective R is less than 1 even before evolution. And then there's the pattern that we see. So just a way of organizing, thinking about these things and thinking about what the consequences of such patterns might be. And the main outcomes, the sort of main statements you come out with this basic model is that for the future of severity, if, and this is a big if, severity and transmission are linked, under pessimistic assumptions around this, possible futures are unbounded selection for increased virulence, so it could get really bad, an intermediate optimal of increased virulence, so it could get a little bit more virulent, or herd immunity that blocks and reduces the evolutionary potential. Of course, it's incredibly hard to monitor. It's obviously very important to monitor to try and figure out what's going on, but it's also incredibly hard to do it because we know that people's vulnerability and also even people's propensity for transmission are highly contingent on features like age, immunological status, the degree of care people have access to. And of course, specifically in areas where individuals are less likely to be vaccinated, we have much less of a lens onto what's going on. So, I mean, really what one can say is that what we do know is that more circulation globally does mean more opportunity for variants to emerge, but it's very hard to predict what circumstances are most propitious for the spread of particular variants. So vaccination and non-pharmaceutical interventions will decrease effective viral population size diminishing the impact of selection and the genetic diversity of the virus. So that might reduce the potential of this occurring. But you know, beyond that, it's sort of everything becomes very sticky and very murky and very context dependent. This is a really interesting question as we start thinking about boosters. We know that transmission blocking immunity from mRNA vaccines does wane pretty fast, which would push us up towards this increased virulence, again, assuming that there is this relationship between transmission and virulence. But it does highlight that one important way to mitigate for virulence evolution is to figure out how to get longer transmission blocking immunity, which might be you know, associated with intranasal vaccines that trigger an IgA response. There's lots of really interesting work in this area. Obviously, monitoring the future of severity is going to be a key part of keeping an eye on this. And this is you know, hard to do, but certainly so important that one should be trying to do it. Tracking infection fatality ratios stratified by age and immunological status is, of course, going to be important. And developing, I think, clinical studies of within-host viral dynamics might actually, you know, give us one way to start pinning down what these trade-offs might look like. I think this is also somewhere where evolutionary ecologists have some helpful things to contribute. And I won't go into this in detail, but we did some thinking around how you might end up with basic ecological principles determining how the virus was spreading within the host. I think another direction one can go for thinking about this is to spread out to consider the broader comparative context, to think about how virulence and transmission are linked across the coronaviruses that we have data from. So there have been 3 coronaviruses spilling over in the last 20 years. And we do have, there are interesting contrasts and similarity in the biology around this. Interesting within host models, for example, of these 3 coronaviruses, and some suggestive patterns like the fact that it seems like MERS-CoV, Middle Eastern Respiratory Syndrome, the coronavirus that somehow never seems to have made it, never really became a pandemic, even though it regularly spills over from camel populations into human populations, is associated, it's a great pathogen of camels, possibly because the receptor it uses, the DPP4 receptor, is highly concentrated in the upper respiratory tract of camels. Whereas in us, that same receptor is concentrated in the lower respiratory tract. So it might be that MERS-CoV is just stuck in this place where it has to be, it cannot get away from being super virulent in us as a result of this receptor that it uses, which makes it a fantastic infection, like a very mild asymptomatic, highly transmissible infection in camels. So I think all these pieces might provide clues on another lens on thinking about how virulence and transmission might be linked. And this, just to end by thanking you all very much. The paper that we wrote on this topic is there, and then there's a couple of others we've been thinking around in this space. Questions to Ian?
Thank you, Jess. Thanks for a great presentation. I was having trouble there finding the unmute button. Okay, we have time for questions. I have a question, unless somebody else wants to chime in. We'd love to see some hands raised, and feel free as well to share your perspectives in the chat box. Should I just go ahead and ask Katya, thank you. You're saving me. Thank you.
I can follow up after your question.
Okay, I'll just ask my question. You know, some patients have gastrointestinal symptoms, diarrhea, things like that. And, you know, we all know that SARS-CoV-2 can also be monitored in sewage. And so I was just wondering if you have any thoughts about, or if you know anything about the extent to which fecal oral transmission occurs, what that might mean in the context of some of the modeling that you guys are doing. And also just back to the, you know, the slide that you had at the beginning with the global vaccination coverage, you know, might this be a different transmission route in low hygiene settings and maybe in low income countries, things like that? I'm just wondering if what's known about that, what you guys know about it, or maybe others on this call might know about that.
So it's very likely that others on this call know more than I do about this. I do have the sense that the degree to which fecal oral transmission is occurring, it doesn't seem like there's strong evidence that that's an important part of transmission anywhere that I know of. There is this fantastic evidence from a porcine coronavirus that switched its receptor. So it went from being a respiratory tract infection, and I think No, it was a very severe gut infection, and it went from to being a mild respiratory tract infection quite suddenly. So, I mean, another thing to sort of keep your eye on is that these things might not be fixed. They might be able to move around in terms of their receptors, which might do really funky things to the virulence transmission relationship. And I think Omicron, you know, again, it's very hard to say whether Omicron is or isn't more virulent because of the background of infection, but it certainly does seem to be using a subtly different infection route. So it's using temperus more, it's not so reliant on ACE2. Ian, did I say anything silly?
No, Jess, I think that all makes a lot of sense. And I think if you just think about the potential for transmission via the respiratory pathway versus the fecal oral pathway, I think given that transmission is still rampant, I think that any sort of selective shift towards SARS-CoV-2 or towards the fecal oral route being more important for SARS-CoV-2 transmission, if that were to occur, I think that would become more of a concern as we get closer to the goal of getting it under control. But I think for now, there's so much potential for transmission via aerosols and things like that, that's going to remain the most important transmission pathway for at least the near future.
Steve Stearns, did you want to add to that? It looked like you had your hand up, or was it a new question? Couldn't hear you. So
I didn't unmute myself. It's a question. So, okay.
Okay. Well, if I could just ask one quick follow-up, what do we know about? I mean, maybe others on this call know something about this, and what do we know about transmission and other species, and you know, some of the other potential reservoirs? Could it be? Is it necessarily aerosol transmission, or could it be fecal oral on some of the other species? I'm just curious if anybody knows about that.
I do not. That's an interesting question, though, because we know that transmission is happening in other species. And I think I read somewhere that prevalence among white-tailed deer in the US or something was just off the charts. So I would be interested to find out what's actually driving that, but I don't know off the top of my head what transmission routes would potentially be involved there.
Okay.
I think that those are so interesting, right, Charlie, in terms of, you know, trying to understand spillover and how it works. We have all these examples into other species for whether when and how those stuttering chains translate into, you know, massive fulminant deer infections. I think it's really something it would be wonderful to have more data on.
Okay, we have Katya, and then let's go to Steve Stearns, and then we'll work through others who have their hands up. Go ahead, Katya.
Yeah, thanks. And great work, guys. So I'm wondering, in terms of actually kind of what's going into the model, is it that, so Jess, you presented early on, you know, this classical trade-off, right, with higher virulence, kind of higher transmission rate, but a shorter transmission time, either because of, you know, host mortality or, you know, potentially kind of, you know, symptom development, which results in isolation then, you know, or so I'm wondering, you know, in terms of previous infection as well as in vaccination, you know, there's been, you know, discussions about whether symptoms actually upon a secondary infection or a breakthrough infection result in symptoms that occur earlier, right? Because they come as a response of as a result of the immune response, right? So I'm wondering whether you're considering kind of vaccination, you know, just kind of lowering the transmission rate potentially, or totally abrogating the transmission rate, you know, or whether you're incorporating kind of this effect of symptom development maybe occurring at a different time, you know, in vaccinated versus unvaccinated individuals, or previously infected versus, you know, uninfected individuals.
So I'm going to let you
Yeah. So in the model that we're using, we assume that the timeline between infection, transmission, and then the end of transmission is the exact same for all individuals, regardless of their vaccination status. Mm-hmm. But you raise a really good point that, especially in the current world, natural immunity is just a huge factor in everything that's going on. And especially I think more and more we're seeing, well, the landscape of immunity is getting more complex. And I would guess that probably the majority of individuals now in the US don't just have one source of immunity. It's some combination of primary vaccination, booster, and maybe some natural immunity thrown in there as well. So I think the timeline to transmissibility is certainly something that needs to be considered in any sort of follow-up to the study. Thinking about what that might actually do to virulence evolution, I think anything that makes symptoms flare up faster relative to the start of transmission is going to decrease transmission potential, presumably, right? Because you start having symptoms, you take a test, and if you test positive, hopefully you're quarantining. So that could be another maybe added benefit of vaccination, even if it doesn't reduce symptoms or transmissibility. If it makes that cue for quarantine pop up earlier, that would definitely help things out.
Great, thanks.
Okay, Steve Stearns.
Yeah, both of you. I know that you've both been concerned about policy as well as about biology. And one of the take-homes from your paper, which I read before I joined this, is that maybe the best thing that we can do to prevent virulence evolution is to vaccinate everybody on the planet. And it appears that the momentum to do that has been dropping. And I wonder if you have ideas about how governments with resources can be motivated to vaccinate everyone on the planet, which, if Charlie's ideas about oral fecal transmission are correct, becomes even more pressing.
I mean, it's very straight to the hardest question there, Steve. The question of how essentially, it would be, one could argue it is in the benefit of every country to distribute excess doses. I was part of a policy exercise in the UK thinking about, whether the UK should be deploying extra doses that they had in other parts of the world. You don't have infinite doses, and it's sort of really hard to decide where you should put the doses because of surveillance is so terrible. Like, there's all these sort of nuances in how the actual policy applications might occur. But the general punchline is that, like, one of the things that we have observed is that the variants of concern that we've seen have not tended to emerge in highly vaccinated settings, particularly, right? They've all emerged before that, so there's definitely no downside from deploying them. How one makes these long-term gains and long-term interests and moves the tragedy of the commons into the background is, I think, a very, very hard question that I'm sure many people on this call have deeper thoughts than I do about.
Yeah, and one follow-up thought to that is that if we're thinking about sort of the global benefits of high vaccination coverage in the context of virulence evolution, those benefits are going to come from transmission suppression. So I think that has to be factored in if we're thinking about how to actually achieve transmission suppression globally. Then I think we either need to be thinking very big and not thinking just how do we get first doses into people's arms? How do we set up pipelines to provide continuous access to vaccines, which obviously there's some huge logistical challenges with that. That's a very lofty goal. But I also think the alternative to that is thinking about how do we, in the long term, shift our thinking from COVID vaccines to purely preventing disease and put transmission blocking effects at the top of the list of priorities, equal to disease reduction effects. Maybe that's developing or modifying vaccines, but I think that if we're thinking globally, we might have to adjust the vaccine development strategy to really match the global goals. But again, that's very lofty, and this is a difficult problem that many people have been trying to solve for a long time.
Would anyone else like to weigh in on this important question? Okay, let's move on then to the next question. It looks like Pere Joan Cardona, you're next.
Hi. I wonder why you have linked virulence with dissemination. Because in fact, when we have looked in the, well, there are several authors that have proved that in symptomatic or asymptomatic patients, they have the same concentration of virus. So it appears that virulence is not linked to the concentration of the virus, and thus there is no relation between virulence, presumably should not be relation between virulence and dissemination.
That's an incredibly knotty question. I mean, I think that's a great point, right? Which is that how do you translate viral titer into movement between individuals? Whether things like coughing might mean that even though I have the same viral titer as you do, I'm actually someone who's more prone to transmit it. I think we're curiously bad at putting all those pieces together. I mean, partly because transmission is never observed, and this has been one of the battles throughout this pandemic, is figuring out what context, what people, the question of the importance of super spreaders for this particular pathogen makes absolutely manifest what a weak handle we have on what enables transmission. But certainly, yes, I mean, there are some subtle differences, right? There's differences in titers between males and females, at least at some stages, and how the time course plays out relative to the timing of symptoms. You can certainly imagine scenarios where these links might happen, but I think we're at the start of building those maps.
Mm-hmm. And I think the other important point is that we are thinking about transmission rate as a population average, right, amongst a certain immune class, right? We know that asymptomatic infections have always been really important. And if you try to model, basically, if you make the transmission dynamics super complicated in the model, you lose a lot of power to actually get these evolutionary insights, and you end up having to make a lot more assumptions. So it was very intentional from the beginning that we didn't break out the asymptomatic class, and we just interpret the transmission rate as encompassing all of the complicated dynamics that lead to a certain overall number. Yeah.
Okay, thank you.
Okay, great. Bernie, are you there? Would you like to ask your question? Thanks for joining us today.
Great, thanks, Charlie. I wanted to thank the speakers for a fascinating talk. I have a question about the fact that there's two sides to virulence. There's the virus and then there's the host. And I always figured that the initial virulence of the virus was more a function of its mismatch to humans than anything else, and it's been adapting to humans. But to really understand this sort of virus, I suspect that it might be important to study more what it does in bats, difficult as that may be. But I guess my question comes from that other side of virulence, that is the hosts. We're coming up on 1 million deaths in the US now, and we know that morbidity to the virus is reasonably heritable. So what can we say, if anything, now about the evolution across the relatively long-term evolution of adaptation to the virus? I mean, it's mostly older people, so that might make it not matter so much. But I think that the strength of selection might be strong enough to have some effects. Can we say anything about that, do you think?
Maybe just to first engage with your first points, which I think are really, really interesting. So the question of how, one of our colleagues, Ben Rice at Princeton, pointed out that it's actually kind of surprising that we only have 4 endemic coronaviruses, right? If you think about it, because we've had 3 spillovers in the last 20 years. So if you kept up that rate for 100 years, we should have at least 15, but we've only got 4. And it might be that it's just a very hard needle to thread spilling over for the reason of virulence, but also for other reasons, which will include the fact of a cross-protective immunity, so if there's any shared adaptive immunity amongst them, but also things like CpG ratios, C's followed by G's within the viral sequence, are something that make things very detectable by our innate immune system, because we tend to have very low levels of such things. And so it seems like, for example, MERS-CoV gets zapped pretty fast by our innate immune system, or detected, because it has a high rate of these. And SARS-CoV-2 has a very low rate of CpG, compared to other coronaviruses and other viruses, in fact, and had it right from the start. And so the idea there is that it might have been selected within, just by chance, it got lucky, it was pre-adapted in that dimension because of selection within bat populations. So I think, so on the one hand, I think absolutely we need to better understand these other host populations. On the other hand, I think what it takes to thread the needle and become a human pathogen is going to be a multidimensional thing, right? It's both, the virulence is clearly an important part of it, but so are many other pieces, like slipping the net of innate immunity. So I think there is some study, and I just don't know this work well enough, so someone else on the call might know it much better, but Pierre Enar and others have looked at a footprint of previous coronaviruses on our population. So have shown, you perhaps know this work better than I do, have shown signatures of selection from previous spillover events. You know, it's one could probe that, certainly. I think that the fact that the major impact really is in older individuals is likely to, as you say, make this less of an issue. But you know, one could certainly write it down. Ian, what do you think?
Yeah, I think that there's a really interesting paper from Jenny Levine and Otter Bjornstedt that came out, I think, around a year ago, about the long-term sort of demographic shift that's expected with COVID, in which as people are exposed, especially young people, and they build up this sort of immunity, you could see a decrease in severity over time, not because of anything to do with viral evolution, but purely because of exposure and this sort of buildup of immunity. And I think that when you layer the evolutionary processes on top of that, I think it's actually going to be quite a while before we can get a really good grasp on how the virus is actually going to adapt to humans, and then beyond that, how it might adapt in the future. Like you said, there's this sort of weird disconnect, especially at the beginning of the pandemic, where virulence is a product of the human and viral factors, and there's probably some mismatch in there, and the virus was certainly not optimally adapted at the beginning. So I think it's just going to take a long time for all of these factors to convalesce, to get to a point where we can even begin to get a clear picture of whether or not there's a trade-off, and where any optimum virulence strategies might be, in order to get a accurate picture. I think everything that we did in this paper, in many ways, it was pandemic science. We were trying to get the back of the envelope answer as quickly as we could. And this is certainly not the end of the virulence evolution story with COVID.
Thank you.
Great, thanks. Meredith, do we have any questions from the chat that we should ask?
Yeah, we do. So we've got a couple of questions in the chat, and feel free to keep popping those in there or raise your hand to join the conversation. But a question from Alan Kugel. Alan asked, how would previous exposure to the virus differ from vaccine effects? Would both act the same?
So I think there, so, you know, most vaccines just have the spike protein in them, and the virus is obviously the whole thing. So your immune system is seeing something different, and it's obviously the experience of being infected will be different, and that will presumably shape how immunity shakes out. There's been a lot of really interesting evidence suggesting that the mRNA vaccines, at least in early phases, seem to have to result in higher protection from infection. Is that right? I think that's right. Than natural infection. There are, of course, all of this shifts and changes the moment you start thinking about many rounds of previous infections. So, you know, whether you've been infected and been vaccinated, or vaccinated and infected, and how these things play out, I think can lead to a very complicated suite of reactions and responses. Omicron, there are some lovely trials that are beginning to look at these different pieces. And there's also this really interesting piece where the duration between, so it looks like, you know, you had your first dose and your second dose, and the interval separating the two, the longer it was, the better protection you got. But maybe too long would not be good, right? So there's another sort of complicated piece associated with the timing, which presumably also plays out for the virus. Ian?
I think that about covered it as much as one can cover the workings of the immune system in a couple of minutes. But yeah, I think that the other thing to think about is the route of infection, because obviously a vaccine is a shot in your arm. When you get COVID, it's spreading all throughout your lungs. So I think the type of antibody protection that's elicited from both of those is interesting, but especially as you mix up vaccinal and natural immunity, it gets complicated fast.
Okay. Terry Taylor, do you want to ask your question? Great to have you here today. Thank you.
Thank you very much, Charlie. And thank you for that excellent and very interesting presentation. I'd just like to explore a little bit more on what's in the policy area, in connecting your excellent work and other colleagues to policy. It made me think of Francis Collins's remark I heard in an interview he made just before he left the National Institutes for Health, the US National Institutes for Health. And he said, he was asked, what area of study do you wish the NIH had undertaken in terms of research in relation to, the question very much in relation to vaccine take-up. And he said human behavior. And so he said that was the one area where he wished more work. I wonder if you thought more about how in the next step of your work, because it seems to me that's the important area, you're bumping up against this barrier of trying to reach out. Vaccination is one of the actions. And maybe in some areas, one almost think about it in the US context, we're reaching a limit on vaccine take-up, for example. And maybe there's room for a multidisciplinary group to get into this particular area, to connect your excellent scientific work with the other scientific work about human behavior, in particular in relation to evolutionary theory. I think there's a great opportunity for bringing people together to take this forward. But I think it's so important to get to this, I was going to say last stretch, I'm not sure that's the right way to describe it, but I think you know what I mean. Thank you so much for your work.
Thank you so much for those comments. And I absolutely, there is, you know, so I worked on measles vaccination a lot before this, and rubella vaccination. And back in the before times, you know, we would talk endlessly about how important vaccine hesitancy was and the absolute urgency of bringing in social sciences and those perspectives. And certainly communication around vaccination has in some ways been extremely disappointing. We have this sort of life-saving tool, which people are extremely hesitant about. I think there is a gap, you know, there's another gap, which is perhaps closer and links to some of the things people have been saying about transmission and links to your points about behavior as well, is that we have a very good understanding of the medical consequences of vaccination from an individual protection, but we often, the piece that we miss is the piece to do with transmission. So there's a kind of individual level studies or population level studies linked to kind of prevalence of things, but the piece that connects the two, the study of, you know, what vaccines do in terms of transmission, etc., is I think curiously unfilled in many settings. I mean, partly because it's very hard. But it's obviously really important, and it's really important in particular in the context of the feedbacks linked to evolution, right? It is exactly this population level consequence of these individual facts and the feedbacks between them that mean we are in this world where we need to think about these long-term things. Ian.
Steve, did you want to add to that, or is it a new question?
No, I'd like to add to it. I'd like to suggest that we give tax breaks to people who get vaccinated.
Yes. Big ones.
Substantial.
Roosters too. Cynthia, you had a question in the chat. Would you like to ask your question? Actually, before we do that though, Ian, did you have anything you wanted to add, or have we? Yeah. Okay. Anyone else want to add to that, those points? Okay, Cynthia, and then we'll move on.
Yeah, thanks. The presentation, the discussion brought up so many things that for us to think about, and it made me think about the following. Do we know anything about, say, in China, where people were exposed to SARS-CoV-1, and does that increase or decrease the vulnerability to SARS-CoV-2?
So I have a very loose memory of a paper I read on this, which I think showed, of course, we don't looking at, was it neutralizing antibodies, that you actually had neutralizing antibodies if you'd had SARS-CoV-1 infection to SARS-CoV-2. But I'm not entirely sure. I need to go back and look at that. I think there's certainly no evidence that of, a sort of antibody-dependent enhancement or really negative outcomes. And I think it's also worth noting that, not that many people, unless there was a huge raft of asymptomatic infections we didn't see, not that many people got SARS-CoV-1 very happily. Because symptoms and transmission seemed to overlap so much, it was relatively easy to shut it down. Relatively easy. It took heroic and wonderful efforts, but it was shut down.
Thanks.
I think that question does raise an interesting point, though, that if we think about coronaviruses beyond SARS-CoV-2, like there are, is it 2 or 3 endemic, very mild coronaviruses that were transmitting in humans? So I think thinking towards the future, there are going to be some really interesting evolutionary and epidemiological questions to be asked about what COVID transmission as well as COVID vaccination does to those other viruses, because no one virus is operating in a vacuum. And I think I am probably not as on top of this as I should be, but I think people are probably going to start doing some really interesting stuff with that sometime soon.
Good point. Okay, Tierney O'Sullivan, are you still there? Thanks for being here.
Yeah, can you hear me?
We can hear you.
Yeah, awesome. Yeah, really great talk, and I really enjoyed kind of hearing everyone pick your brains this afternoon. So I just had a question, I guess, given kind of the topics that you're trying to model specifically with like transmission versus virulence and immunity with vaccines. Is there anything that would be really helpful to include in vaccine trials that would aid with modeling? So like, for example, if you tested everyone regularly to see if they had asymptomatic infection versus just symptomatic COVID and stuff like that, how that would be able to inform your sort of modeling better.
I mean, I think that would be wonderful. And I think that's precisely what you need, right? I mean, the vaccines do exactly what the trials were designed to test, which is block symptoms and serious disease. But we didn't, and I mean, a lot of it was happening. It was pandemic science. It was incredible. It was extraordinary. Like the relevant vaccines was extraordinary, but that is precisely something that wasn't measured from any of these. And I think that would be number one on the list. Are there other things, Ian?
Yeah, I think one very interesting thing is when we're thinking about transmission, there are really 2 types of transmission that could be affected by vaccination. One is the odds that you become infected when challenged, and then the other is the odds of transmitting to someone that you contact when you are infected. And I'm not sure if the human challenge studies would be able to address that, but I know that Mark Lipsitch wrote a really interesting article where he proposed a human challenge studies where the participants played a chess game with each other or something like that, and one person was infected to simulate like the natural process of infection. And again, I think that he proposed this as like a pandemic science solution to getting vaccine results fast. But I think studies like that could provide like the really detailed type of data that you need to tease apart all of the different factors affecting viral evolution. But you open the ethics can of worms with that. And I think that for the next pandemic, hopefully we will be a little bit more agile when it comes to thinking about how to conduct vaccine trials in an emergency situation. But I mean, as Jess said, the efforts as they were heroic, and it's this unprecedented problem. And it's amazing what the world was able to pull off so quickly as it was.
Awesome, thank you.
Okay, we had a question in the chat from a while back by Fernandez Gonzalez Candelas. Are you there, Fernando? And would you like to ask your question?
Yes, I'm here. Well, thanks for the great talk. In light of the evolution of the immunity, by the waning of immunity, how do you think that will affect your model and your results on the evolution of virulence in the pathogen?
So it's immunity to what is, of course, the question, right? If it's waning immunity to infection, but it's not waning immunity to disease, then we're in a different situation than if it is. And if those two map perfectly to each other, perhaps Ian, do the hard answer.
So I think the evidence that we're seeing right now is that protection against infection is waning faster than protection against disease, which in the plot that Jess showed would push you to the left towards worse outcomes for virulence evolution. The question is, like, how do we get further to the right, given that waning is happening? And boosters are sort of the obvious answer to that, because you get boosted, you get that temporary robust protection against infection again. But the other piece to the puzzle is natural immunity, right? Because we know that people are becoming infected even after they've been vaccinated. So natural infection might push the outcome to the right in that plot again. But of course, natural infection, there are all of these risks associated with that. So I think that to basically sum up the problem that's at the end of this entire study, it's how do we get further to the right on that graph? How do we deal with waning and push ourselves towards the best scenario for mitigating virulence evolution? And I think that's the big open question right now.
Okay, great. We have Sujit Pujari has her hand up. Would you like to say anything? Would you like to ask a question?
Yes, sure. It was a nice talk. Thank you so much. My question is not really a question, but during the pandemic, we saw some few products that were coming, a nasal spray based on RNAi-based technology. So what do you think about that? Like, we didn't see anything in the market yet, but if something like come in the market, do you think that will be effective enough to block the transmission and stop the pandemic at some point?
I don't. So the question is what we mean by stop the pandemic. I don't think we're getting rid of this virus. I think the chances are, and again, who has a crystal ball, I'm not sure, but I think the likely trajectory is the one outlined in the Levine et al. Paper that Ian referenced earlier, where we move towards endemic transmission, but hopefully we're in a space where so there's a rising tide of T-cell immunity in the whole population that protects from disease, and so it's another mild cold. We might be in a world where we need to manage that with boosters or vaccines, if we get unlucky. The mRNA technology, so Ian, I think you're more up to date than I am on this. The mRNA technology is maybe not so good at blocking, or at least I don't know that we've got a really good handle on developing a vaccine that develops IgA immunity, which is the immunity that would block transmission, right? That's sort of based in your upper respiratory tract. Is that mRNA? I forget, Ian, that particular vaccine.
So I think that all of the mRNA vaccines currently in use are intramuscularly delivered in a shot. And I think it's more the IgG response that prevents mostly disease. As far as I understand it, has mostly to do with the delivery route. I believe that there are nasal spray vaccines that use the mRNA technology in development. Those should have a little bit more potential to block transmission. Again, I don't think anyone has a solid handle on how that protection might wane relative to the traditional shot. But I think that like the huge advance that we've seen in technology coming out of the pandemic is these mRNA vaccines. And I think the potential to be able to very quickly adjust the vaccine to match whatever the dominant variant is amazing. So I think if we, the very royal we here, the pharma companies can figure out how to both track variants and prevent transmission at the same time, that's going to be a true game changer.
And I mean, I think just to say one more thing on that, right, Ian, I think that the evidence so far is it's kind of there's at least one, I think it was a mouse model study that suggested at least that an Omicron specific vaccine didn't do that much better in terms of protection from disease, right? So there's also the interesting question of like, when should you jump the gun and go for the boosters, and when are you doing good enough? Like, how do you manage those trade-offs?
Yeah. But and the other piece to that is that's in some ways that's an N of 1 example of a variant specific vaccine, right?
Absolutely.
I think that this is, I mean, it's a whole new field of these sort of not exactly precision medicine, but rapid vaccine development. And yeah, it'll be really exciting to see what comes out of that in the near future.
Okay, great. It looks like Steve Stearns has taken up Meredith's challenge for one final question. So we'll turn it over to you, Steve.
Not a question, it's a comment. I think it's a cosmic irony that a mother in East Africa decided that her daughter should not be vaccinated and that she should get all of the childhood vaccine diseases, and that daughter would then turn out to be one of the leading vaccination experts on the planet, which makes me think that the sociology of evolutionary medicine has in it a component that we could call the developmental origins of adult career choice.
That's a final comment from me. Stephen Stearns wrote me a handwritten note for the first paper I ever wrote, and maybe that also led to the career.
Okay. Well, thank you both very much. Thank you, Ian. Thank you, Jess. Thank you, Charles, for doing the introductions. Thank you, Meredith, and thank you everyone for tuning in. It's great to see so many people at Club EvMed. Stay tuned for our next set of talks that'll happen early in the summer, right up until the conference, International Society for Evolution Medicine and Public Health Conference. And then we'll take a little break and start up again in the fall. So thanks, everybody. Great to see everybody.